achieved through the presence of antibodies to that disease in a person's system. Antibodies are disease-specific proteins produced by the body to destroy toxins or disease-carrying organisms. Your immune system is broken down into an innate and acquired forms. Your innate system is what you're born with, and the acquired part is from exposure to pathogens. Your acquired system is further broken down into natural immunity, which is obtained from getting sick, and artificial immunity, which is obtained from vaccines. Each of these components has both an active and passive form.
Before we move on to talking about passive versus active immunity, there's a little bit of review needed for the different types of immunoglobulins, as they are the main players in our immune system. So just going through the big four immunoglobulins. First, we have IgE. It is a monomer, and it's mostly used in the allergic response. So once an allergen enters your body, the IgE binds it, and then it also binds mast cells and basophils to trigger an immune response. And they do that by releasing histamine. Next, we have IgG. This is the most ubiquitous immunoglobulin. It is also a monomer, and it is found in 4 forms, and it's also the only immunoglobulin that is capable of crossing the placenta. Next we have the IgA, which is a dimer, which means that there are 2 monomers attached at the ends. IgA is our first line immunoglobulin. It's mostly found in our mucosal membranes and it has the most exposure to allergens. So you'll find it in places like the gut, the respiratory tract, and the urogenital tract. And it also can be found in bodily secretions such as saliva, tears, and breast milk. Lastly, we have IgM, which is the biggest of the immunoglobulins. It is a pentamer, which means that there are 5 monomers attached at a central location. And IgM is kind of the first immunoglobulin produced. So it's found on B cells. And when you first get an infection, the B cells are able to churn out large amounts of IgM. And that is generally a good indication that someone has had a recent infection. So now that we've talked about the different antibodies, we can now start talking about passive immunity. Passive immunity basically means that someone receives antibodies that are pre-made. They don't have to make it themselves. There are 2 ways to get passive immunity. One, you can get it naturally through your mother as a fetus, and a baby when it's born does. And 2, you can have it injected, which is the option that is available to most adults. So, number 1, how do we get immunoglobulins naturally? There's actually 2 ways. One, you get it in utero. As we talked before, IgG can cross the placenta. So in utero, you will get all of your mother's IgG antibodies that she has formed. And this will protect you for the first few months of your life. Secondly, once you are born, you can get IgA antibodies in breast milk. Remember, IgA is the immunoglobulin that is found in secretions, and that's why you can find IgA in breast milk. So once you're an adult, you don't have the options of the naturally made antibodies, so we have to have injected antibodies. So there's basically what happens is that the antibodies are formed in a different organism, human or horse, and then they are harvested from that organism and then injected into an immunocompromised person so that they can fight off whatever infection that they are at risk for.
This brings us to active immunity, the process which creates long-term protection against many pathogens and diseases. Active immunity results when exposure to a disease organism triggers the immune system to produce antibodies to that disease. Active immunity often involves both the cell-mediated and humoral aspects of immunity, as well as input from the innate immune system. Just to note, The innate immune system is present from birth, and it protects an individual from pathogens regardless of experiences or exposure, whereas your acquired immunity arises only after an infection or immunization, and hence it is acquired during one's life. Initially, with a primary exposure, B cells and T cells are activated by a pathogen, resulting in memory B cells and T cells developing. Throughout a person's lifetime, these memory cells will remember each specific pathogen encountered and are able to mount a strong secondary response if the pathogen is detected again. Exposure to the disease organism can occur in two ways. It can be through an infection with the actual disease, which results in natural immunity, or introduction of a killed or weakened form of the disease through a vaccination, which provides artificial immunity. Either way, if an immune person comes into contact with that disease in the future, their immune system will recognize it and immediately produce the antibodies and effector T cells needed to fight it. This makes future infections either mild or inapparent. Artificially acquired active immunity is where immunizations come into play. This can be induced by a vaccine, which is a substance that contains antigen. A vaccine stimulates a primary response against the antigen without causing symptoms of the disease. They basically teach your body how to defend itself when germs such as viruses or bacteria invade it. They expose you to a very small, very safe amount of a virus or bacteria that have been weakened or killed. Basically, your body learns to recognize it and attack the infection if you are exposed to it in the future. There are 4 types of vaccines that are currently available. First, we have live virus vaccines, which use the weakened or attenuated form of the virus. An example of this is the MMR vaccine or the varicella or chickenpox vaccine. The second form is the killed or inactivated vaccine, which are made from a protein or other small piece taken from a virus or bacteria. The flu vaccine is an example of this. The third type are toxoid vaccines, which contain a toxin or chemical made by the bacteria or virus. They make you immune to the harmful effects of the infection instead of the infection itself. Examples of this is the diphtheria or tetanus vaccines. The fourth type are biosynthetic vaccines, which contain man-made substances that are very similar to pieces of the virus or bacteria. The Hib vaccine is an example of this.
Next, we're going to talk about herd immunity. When a large percentage of the population is vaccinated, the spread of disease is limited. This indirectly protects unimmunized individuals, including those who can't be vaccinated and those for whom vaccination was not successful. This is the principle of herd immunity. In successful herd immunity, in diseases that spread from person to person, it is more difficult to maintain a chain of infection from when much of the population is vaccinated. As the number of those vaccinated increase, the protective effect of herd immunity increases. For some diseases, herd immunity may begin to be induced with as little as 40% of the population vaccinated. More commonly, and depending on the contagiousness of the disease, vaccination rates may need to be as high as 80 to 95%. This percentage is called the herd immunity threshold. Conversely, in unsuccessful herd immunity, when only a small percentage of the population is vaccinated, the risk of a disease outbreak is greater than if many are vaccinated. The unvaccinated members of the population are not indirectly protected, and each community member has a higher risk of becoming infected. So lastly, we're going to talk about the vaccination schedule as per CDC recommendations. It is important to note that the CDC actually revises its recommendations every 12 months. So we're going to try to break this down for you as quick and painless as possible, as we know that the vaccination schedule can be quite confusing. So when the baby is born, at birth, he or she first receives the Hep B vaccination. At the 2-month checkup, the baby receives a whole host of vaccines, including Hep B, DTaP, which is diphtheria, tetanus, and acellular pertussis, PCV, which is pneumococcus, the rotavirus, influenza B, and the polio vaccine. At 4 months, the baby receives the same vaccinations, the second doses of all the same vaccinations at 2 months, except for the Hep B. The 6-month visit is the exact same as the 4-month set. At 12 to 18 months, the baby or child now receives Hep B, the DTaP, the PCV, the Hib, and the IPV. In addition,
These children now receive the MMR vaccine, which is the measles, mumps, and rubella vaccine, the varicella vaccine, which protects against chickenpox, and 2 doses of the Hep A vaccine given 6 months apart. At 4 to 6 years of age, the child now receives another dose of the DTaP and IPV vaccines, as well as the MMR and varicella.